Thank you so much for joining me for today's lecture related to non-clinical studies that we are going to add into our CTD or Common Technical Document when we are going to make any type of application such as new drug application, new drug submission for Health Canada, for IND investigational new drug, for FDA, or for CTA clinical trial application, as well as for MAA marketing authorization application for Europe. And of course, the CTD format is used for several other applications for the registration related, I would say, content that you are going to submit to regulatory authorities in different countries in the world. Allow me first to remind you that the structure of the CTD or Common Technical Document is the one that we follow from the ICH M4, which is International Council of Harmonization. You can find all of the guidance documents from the ich.org at, again, the website, ich.org, of course. And you can always click on the M for the section M standing for multidisciplinary, and then to find ICH M4, where we'll be able to see the structure required in order to prepare our submissions in the CTD format. So on the top left side of the screen, you can see different modules. And again, this one is about the, I would say, new drug submission. But again, module 2 through module 5, they are all universal and they are being used in any type of submission in CTD format. So when we talk about non-clinical studies, we are not just talking about module 4. We also have in module 2 a module or a sub-module where we have to add all of the information that we have related to the safety and efficacy of our new product tested in animals or animal models. This is why first I'm going to start talking about module 2.4, which is, again, if you go to, again, different modules, as you know, M1 is for Canada, and we already went over the different sub-modules today. I'm going to go over module 2 and module 4. So module 2 does have several sub-modules. The one that we are going to talk about now is a sub-module 2.4. So module 2, sub-module 4, and when we open this again, a sub-module, we're going to see that we'll have to add in the space or in this sub-module information related to the clinical or non-clinical studies. So what should we put into this sub-module? So let's go over what we add here. Here I am showing you the content of the information related to non-clinical trials in module 2.4. So let's go over again the table of contents, what we should add here. So as you see, this is a well-formed document. This example is coming from a publicly available document that you can find on internet and on different regulatory authorities where they share the information related to the drug submissions. So as we see, we will have to follow this table of content for module 2.4. So after going over the overview of the non-clinical testing strategies, we talk about the introduction, the rationale, the non-clinical development plan. Then we talk about the pharmacology, mechanism of action, secondary pharmacology, safety pharmacology. Secondary pharmacology means, I would say primary pharmacology is when we are dealing with, again, the effect of the drug. Of course, this is going to be pharmacokinetics and pharmacodynamics, and if applicable, pharmacogenomics. But this is related to the effect of the drug. However, when we talk about secondary pharmacology, we are looking into the effect of the drug on non-target organs or tissues. So here we talk about secondary pharmacology if we did, of course, perform tests for learning more about the effect of the drug on the body or what the body does to the drug. So when we talk about pharmacology, of course, we talk about pharmacokinetics, where we are looking into the absorption, distribution, metabolization, and excretion of our drug in different animal models. Obviously, this section is going to be followed by toxicology. So we are going to give the one single dose of a toxic toxic dose to the animal model, and then we are going to look into the effects on the animal, and then we are going to have repeated doses. So it's not just one dose that we give, we are going to give several doses with different intervals depending on our strategy for doing research, and then we are going to get the results and add them into this section. We also look into genotoxicity, carcinogenicity, and we're also going to look into, if applicable, the evaluation of tumor findings and embryo fetal toxicities. Then we continue with reproductive and developmental toxicologies. So, of course, I'm not going to go over each and every of these, the content, because, again, we do have more information related to, for example, section or module 4.2.1, etc. But I'm now going to just show you several types of content that we have in our module 2.4 content. So as you see, we have the introduction. It's about a few paragraphs. Then we talk about the rationale for use of the drug. Then we do add a few paragraphs related to the non-clinical development program. So we talk about the strategy, what we have done, what we wanted to look at when we were doing studies, of course, in animal models. And then we are going to talk about the pharmacology, mechanism of action, secondary pharmacology, as I already explained to you. And then we look into the safety pharmacology, where we are looking into adverse drug reactions, any untoward medical occurrence experienced by of course, the animals who have been exposed to our experimental drug. As you see also very well, we are not adding too much details into this document, because in module 2 we are giving information which is much more a summary of what we add in module 4. Again, for the sake of this presentation, and of course for the first half of this presentation, I am talking about the content of module 2. However, I'm also going to continue talking and showing you the content of module 4, which is for non-clinical studies. So continuing with the content that we add in module 2.4, we can again give examples of the findings in pharmacokinetics. So here, for example, you see it's the oral administration. We may also have some other types of using some other types of routes of administration in animals, depending on our strategy to explore about the safety and efficacy of our product in animals. We also add here a summary of the findings for the absorption, the distribution, metabolization, and of course excretion in animal species. Then we are going to continue with toxicology, showing that when animals are exposed to a dose that may now exert its effects that are toxic, we are also going to talk about that. So here, for example, it was determined that for this specific drug, the dose of 300 milligram per kilogram of animal per day was toxic. And then we are going to again continue giving the summary of our findings as well. As I already mentioned, we will go into details in module 4. In module 2, we just go over the summary, and this document is the one that we prepare for this module. So after single dose toxicity, we are going to talk for I would say one paragraph or two about the repeated dose or multiple dose toxicity. We also do studies on genotoxicity, on carcinogenicity, on, of course, the evaluation of tumor findings, and on embryo fetal toxicities. So, of course, reproduction and developmental toxicology as well. But of course, we saw here in a nutshell, again, what is the content in module 2 related to animal studies, which is a summary. So now I'm going to again show you another table of contents for another drug. So as you see here, we are again adding in different, in module 2, different contents. What you see here now is that in module 2 that I already showed you, we can also create sub-modules, such as sub-module, I would say 2, 4, 2, 2, 4, 2, 4, 3, 2, 4, 4, etc. And this is what you can see here in this another example of the content that we put in module 2, submodule 4 of the CTD. So here, of course, this is much more granular, so allow me to go over this content as well. So when I am going to, when I talk about, for example, 2.4, it means that this is this module. And when I'm talking about 2.4.1, it means that here I have to have a folder or a module called 2.4.1. And what goes inside? This is going to be the overview of non-clinical testing strategy, where we can now add different tables, all related to this submodule 241, and of course 124.4.12, and then we continue with another submodule that we are going to create. This is going to be, we're going to call that module or submodule 242. It's going to be about the pharmacology, and inside that submodule we are going to create another submodule. Don't forget that based on the ICH M4 we can only go down to 5 levels of modules, and this is what we call the granularity. So again, here we have module 2, submodule 4, submodule 2, submodule 1, and here we are adding primary pharmacodynamics information. And within this module, of course, as you see, we are reaching 5 levels of granularity, 2, 4, 2, 1, 1. And again, if you would like to find that out, we have to create this module 2.4, which is already there, as I told you, on the top left side. Then we have to create submodules 241, 242, 243, and then we will create also submodules 2411, 2421, 1, 24212, etc. So this is how we are creating the structure of the CTD if we need, of course. And the same thing applies to module 4 and 5, where we are not just having a module as a placeholder for one clinical trial, but we are going to create sub-modules when we are having a higher number of clinical trials, or obviously, for the sake of this presentation, for the non-clinical studies. So for continuing with the content, as you see, we are again going over the details. So pharmacokinetics is going, we are going to have again a brief summary in one submodule. In a document, we are going to have another document for the method of analysis. It will go, of course, into this submodule. We are going to have another submodule called absorption, and we are going to create again submodules within this submodule. So 2433 is going to have two other submodules, 24331 and 24332. In each of those, we are going to add the information related to in vitro absorption and single dose pharmacokinetics. So now if you would like to continue with, again, very long document of 36 pages, you see that this is, again, following the logic of the CTD, which is based on having specific information in specific submodules, now module 1, of course, through module 5. And when I continue, of course, here we can have a list of abbreviations. Then after the list of abbreviations, we are now going to see the content of the module 2, submodule 4, submodule 1, which is called Overview of Non-Clinical Testing Strategy. And the content that you see here is exactly the content that will go into that submodule. Okay. Perfect. So as you see, it's more than here than one paragraph. This is much more in detail. And then when we are going to talk about pharmacology, obviously, we are going to add the information related to the pharmacology in animal studies in another submodule, which is module 2, submodule 4, submodule 2. And here again, we do have other submodules, as you see, and each of those is going to be called the way that it's written here. And we are going now to continue to create well-formed documents containing, for example, this information into the well-formed document that we are going to add into submodule 24211. So as you see, the information that is in this document is coming from different sources. All of those sources are, of course, from animal studies that have been performed. Now you may, again, companies, they may have their own facilities, or they may outsource this activity so that regulatory affairs professionals are going to receive again, information from different sources, various sources, and those sources are going to allow RA specialists to get the information, add the information into well-formed documents, and then to prepare step by step all of the documents that are going to go into each and every of the modules and submodules in the CTD. So coming back to module 2, submodule 4, for the CTD structure for non-clinical overview, we are looking into, of course, the content. And as you see, this is, again, just a summary of the findings that we have to add into each of the submodules. Perfect. So now that we went over this, I would like now to tell you that in addition to, of course, module 2, where we add the summary, we are dealing with module 4. So I'm going to invite you to look into the top left side of this screen, where I'm going to click on module 4. And in module 4, obviously, I am going to see as a placeholder or a submodule 4.1 for the table of contents. Then we have the submodule 4.2, which is about the study. Now here, for any type of study related to the pharmacology, we are having several submodules in module 4. So here, for example, we have the primary pharmacodynamics. We have submodule 4212, secondary pharmacodynamics. Then we have module 4213, safety pharmacology. And then 4214, this is for the, again, drug interactions. Then we can look into the module 422, where we have to add analytes, of course, and metabolization, validation. Here we have the absorption, distribution, metabolization, excretion, kinetics and drug interactions, and other pharmacokinetics studies. And for continuing, we can have information related to the toxicology, where we have module 423, and then you see that you have several submodules. Again, any information that we have related to these, so single dose toxicology, repeated dose toxicology, genotoxicity, carcinogenicity, reproductive development toxicology, etc., we are going to have placeholders for those. So now if, for example, you did perform other toxicology studies related to immunotoxicity, etc., you're going to have a placeholder for these different types of studies. And what you do is that you're going to add, of course, to prepare documents, and you're going to add those relevant documents into the corresponding submodules. Now, coming back to, again, the reproductive developmental toxicology, same here. We are looking into fertility embryology development. We have another module or submodule for embryological fetal development. We have pre or postnatal development, and of course juvenile animals. So that was in a nutshell what we have to add in module 4 of the of the CTD, and now I'm going to go over several documents. For example, let's go over a document that I am already having here. Again, all of your documents are publicly available, and that's why I am sharing this with you or showing them to you as well. So let's go over, for example, content of 4211, primary pharmacodynamics. So where is 4211? 4211, if you look at the top of the screen, is in module 4.2, and then 4211 is here. So this is 4211, primary pharmacodynamics. So the information that I'm showing you here, primary pharmacodynamics, it must be prepared, and then it must be added into this folder. So of course I don't do that. It's just for showing you the placement of that file. Now I'm going to just open that for you, because it's important to know that those documents are not necessarily large documents. In fact, I just showed you again the content, examples of the content of module 2.4 for non-clinical summaries or overview. But when we are dealing with again module 4, we are going to need to prepare several well-formed documents. So they must have again the header, a footer if applicable, and then we need, of course, to add only the content corresponding to the information required for a specific module. So when we are looking into primary pharmacodynamics in animal studies, we are only going to add information related to pharmacodynamics, again related to the animal model and of course the use of our drug, and we are going to add the information into this well-formed document. So for example, here you can see for the primary pharmacodynamics, angiotensin 2 inhibition. So we are looking into again what the drug does in that animal model. We talk about the impact of hypertension, then renal function, and then as you see, this document is not larger than that because we are just adding the information related to pharmacodynamics and primary one. So now you may say, in this case, are we going to have necessarily large documents? The answer is yes, depending on which document you're talking about. We do have, for example, in module 3, documents related to the CMC or chemistry and manufacturing and control. However, when we are looking into making well-formed documents, we will see that in some of these well-formed documents, we are going to add only just a chemical formula, or in another one, just the nomenclature or the naming or the name of that compound. However, in some other documents, we are going to put the full analysis and, of course, with the calibrations, with all of the results for I would say, a procedure that we use in order to show that, to test that our drug is the one that we are claiming. So it will depend on, again, the type of documents. Some documents are just half a page. Some of them, they may be 50 pages as well. So here we are having, again, a document which is just 2 pages, 4211. Now let's go over what is in 4212. In 4212 or module 4, submodule 2, submodule 1, submodule 2, we are having a document related to secondary pharmacology. So here I'm going to show you again a document related to the secondary pharmacology. Here we are again talking about again variability in blood pressure and throat to peak ratio, orthostasis with symptoms, combination counseling, and then effectiveness in various population, etc. Again, as you see, this is just 2 pages of important, of course, required information, but again, it's just 2 pages that we use. Now, let's go over what is in section or module 4213. This is related to safety pharmacology. So let's go over that, open an example, again, publicly available example of this, document. Now we see that we are looking into the safety profile of our drug that has been tested on animal models, and here we are talking about different systems and the effect of the treatment in animals on those systems. So here this is just a one-page document where we are talking about again, safety pharmacology related to the cardiovascular, to the renal, or to the central nervous systems. Now let's go over again what is in the pharmacodynamics and drug interactions. The content must be in this submodule. I'm going to open that information. This is a document that we prepare and add into, again, this module. So what is here again? Let me just close this one, and this is the document that we have to look at. So this is the pharmacodynamics drug interaction. So here, talking about the drug interactions, we tested again different drugs in addition to I would say testing our experimental drug, and we looked into the drug-drug interactions. So interaction between our experimental drug and other types of drugs. So here we are looking into NSAIDs or non-steroidal anti-inflammatory drugs, and the drug-drug interactions between NSAIDs and our experimental drug, and this applies to other types of drugs like diuretics, like again hypotensive drugs, on potassium supplements, on lithium, on digoxin as well. So this document is about 3 pages, and it just allows us to look into the drug-drug interactions, what we found in animal studies. Now I'm going to look into again another document. For example, I can first of all allow me to just close these documents first. Now, for example, you can have 4221 primary pharmacodynamics. So let's go over what we have to put in module 4221. I will go to here 422 on the top left side, 422, and then I'm going to search for 4221. Perfect. So here I'm going to add in 421 primary pharmacodynamics, and I'm going to open this document, and we are going to see what goes into this content. So this is about the pharmacodynamics information from the animals, and don't forget that we also use previous research done by again other investigators as well. We can also refer to previous work done and published as well, in addition to what we can again do for testing our drug. So here that's a 6-page document related to this, and that's not the drug, of course, this drug. And here we are looking into the primary pharmacodynamics. Perfect. And now let's go over what is in the next subfolder. We are looking into absorption. So 4222, this is module 4, submodule 2.2.2, and I'm going to now show you an example of a well-formed document where on the top we have the again the header, and here of course we have the reference to the eCTD section or the submodule, and here we are looking into the absorption. So we talk about the bioavailability of oral drug, time to peak concentration, bioexponential decay kinetics, first phase, terminal phase, half-life of terminal elimination, lack of accumulation, of course here. And again, this is just a 2-page document related to the absorption of this product. So we create this document based on the information that we get from animal studies. We may have not one but several animal models, obviously, and in this case we are going to add all of the information into this document. Once the document is made, we are going to convert it into PDF. We are going to do the quality control of a document, since the conversion from any software such as Microsoft Word as well into PDF may end up eliminating or changing special characters. In this case, we need to necessarily um, uh, quality control or check the content or the text or the content that we added into a document after its conversion to PDF as well, for making sure that no special character like percentages, for example, have been changed for accuracy. And once the PDF is done, we are just going to drag the document and drag it and drop it into the submodule. Then the next module is, or submodule is, about the distribution 4223. So for this one, again, this is information related to this drug. And here we are looking into the binding of plasma proteins, if it's albumin, if it's something else. And then, of course, may also have fasting animals as well. For example, you know very well that fasting can affect the level of the plasma proteins, and if there is a binding of our drug in the to the plasma proteins, it's going to affect its metabolization and its distribution as well. So we look into the binding of plasma proteins. Here we talk about the amount of the distribution, the effect of food on absorption, because you also have food interaction with the proteins, for example, that the food has in the blood, and then it's going to affect again the availability of the drug in the blood, as well as, of course, its metabolization as well. And then we look into the dose. The dose affects how much of a decline there is. So maximum concentration, or the Cmax, comparable plasma concentration following 3 hours. So we look into different conditions where those conditions may affect the plasma proteins as well. We are looking into really a thorough study of the availability of that drug, again, into different organs in different animal models. And again, another 2-page, but very important pages as well. Again, make sure that you, again, double check for the content. Also convert that into PDF, quality control or QC the PDF file, and then we are going to now drag the PDF file and drop it into this module. Now for the module 4224, for the metabolism, we do have again here, for example, we have this document. So here we talk about the conjugation with glucuronic acid, glucuronic metabolite exclusivity, plasma's metabolite proportion, absence of CYP, isoenzymes, again, participations. CYP450 are enzymes that are, again, involved in the metabolization of, again, different food as well as drugs and toxins. And we call them cytochrome P450s. We have more than 17, 18 families and hundreds of subfamilies of the CYP450s. Don't forget that the metabolization is not only by the CYP450s, but this is, I would say, is a major disorder. That's a major family of isoenzymes that are, again, metabolizing drugs as well. Then here we can see that this is just, again, 2 pages of information. We save the information, this page, into PDF, and we drag and drop it into this folder or submodule. Then looking into the excretion, we are going to look into module 425, which is here, okay? And then the content has, of course, the titles like complete plasma elimination, half-life independence, and complete dosing clearance, the primary method of elimination like biliary excretion, excretion of glucuronide metabolite in urine, and then fecal in the feces excretion, or disparities between genders in blood pressure response and clearance. So again, another 2-page document, right to the point, related to the excretion of the again, experimental drug in animal model or animal models. And this document has to be added into module 4, submodule 2, submodule again 2, 5. So 4, 2, 2, 5. And this is the way that we also give the name. So even when you see the way that those submodules are named, this is the standard. So we will need to use the same naming convention for these submodules. The same applies to other modules that I already showed you. Now let's go over the continuing with this. So in module 4226 here, we are again talking about the pharmacokinetics and drug interaction. So here we talk about the volume of distribution, protein binding, metabolism, the metabolic pathway, if of course it's known, primary metabolites, we talk about or add information about the excretion, removal half-life, excretion routes, drug interactions, a mechanism, of course, of that, and then clinical implications. Now we are going to look into, again, effects on other, of course, excretions like NSAIDs, or, and of course, the clinical implications on potassium supplements and the clinical implications, lithium, digoxin, etc., etc. Again, we don't only look into digoxin or warfarin or lithium. It will again depend on the strategy that we develop in order to test our drug or experimental drug in animal models. Okay, so here I'm just giving an example of a specific drug, experimental drug at some time and in the past, and then you see that the information that you need to use in order to prepare all of these documents again will come from different sources, but here mainly from places or organizations or teams that did perform animal studies. Now looking into again 4226, perfect, pharmacokinetics. The information or this document is written, and then it's going to be added into again the submodule 4226. So inside we do have obviously the information related to the pharmacokinetics and drug interactions. So this one again is a 3-page document, and as you see, we are giving specific information related to this topic. Now, you can also have, of course, other pharmacokinetic studies as well. And again, if not, we are now going to start looking into, again, other documents. For example, we have 4231, okay? This is for the toxicology, 4231, which is a single dose toxicology. And here I'm going to show you again one of these documents. So here, as for another drug, we are looking into the dosing. So with the dosing, and don't forget, this is for the toxicology. So we are going to see any side effect, any signs and symptoms related to an overdose. And we are also going to establish what is that dose and what are the effects or side effects of that toxic dose on different animal models that we use. So here we have different topics like minimal therapeutic dose and dosing ranges. We have, for example, toxicology. It's also, again, about the single dose. Okay, you have different tables, approximate conversion factors, smoked and oral for that product, etc. So again, as you see, for almost all of the documents that we are having here, these are all documents that are short, but again, the content is coming from study reports or animal study reports that exist, and regulatory affairs specialists, they use or we use the information in order to write these well-formed documents. Then we can also look into repeated dose toxicology. So this is in submodule 4232, where we will be able to see this document. So here, this is just again a table of the overview of the repeated dose toxicology studies with this specific drug. And again, the information could have been maybe described better by adding some explanation. But again, for this specific document, that's how authors judged that it was sufficient. Then we are looking into genotoxicity information 4232. So here I'm going to show you again a document for genotoxicity. And here again, we are going to write about the information about the genotoxicity, which is only 1 page for this drug. Then we can talk about the carcinogenicity. This is 4234. So module 4, submodule 2.3.4, where we will have the information about the carcinogenicity. Perfect. So we put the doses as well, and we are going to write about the I would say the results of the studies that have been done in different animal models. Now we can also look into the fertility embryonic development, which is in section or module 42351. 4235 is here, and then you see that in on the top again corner of this left corner of this slide, you can see that when you click on the submodule 4235, you have several submodules. So we are going to look into what we are going to add into submodule 42351. This is what we are going to prepare and add into this module. So here we are talking about the fertility, pregnancy, and lactation. And we just add the information here, save it in PDF, check it for special characters, and of course for again final approval. And then we are going to add that into this subfolder. For the embryonic fetal development section again or submodule, we also have this document, which is the one that I'm showing you. So again here it says, for example, non-clinical investigation did, however, identify some possible risk of the drug to fetal and postnatal development at higher toxic dose levels. These larger doses were linked to more late resorption in rabbits, suggesting possible hazards to fetal viability. Don't forget that we look into, again, different toxicological studies as well, because we would like to establish a list of potential adverse drug reactions that, again, our patients may experience. So this is why we always look into this type of information, and we need, again, of course, to have those type of experimentations in our reports. So then we can look into 4236, which is a local tolerance. So let me just go over 4326, which is this one. So with the 4236, we are going to add this information, which is here. So tolerance, dependence, and withdrawal symptoms. So now when I continue, of course, I can see that you have, again, about 1.5 pages of information related to the tolerance. And then we have, again, other toxicological studies. So this one is, again, if in case we do have any other, I would say, toxicological studies, that we are going to add the information here. Now, we also have, of course, literature. So we are going to look into what we are going to add into the literature reference. So here, this is going to be, again, the list of all of the references that we're using. Of course, here, this is written this way, but it will depend, again, on the standard operating procedure that any pharmaceutical company is going to use. So usually, we are looking into, again, formats like APA format, but here, just for the sake of presenting the content, I'm just adding these HTTPs. But again, make sure that we are respecting the procedures or standard operating procedures in any pharmaceutical company where we are working. So in a nutshell, and I thank you so much again for joining me for today's class, we were able to look into, again, the content of Module 2 and the content of Module 4 of a CTD or Common Technical Document. Again, for the submission of many types of regulatory submissions, we looked into, again, different sections. We looked into Module 2, where we looked into the Module 2.4, non-clinical overview. Then we looked into Module 4, and we explored each and every of the, modules and sub-modules and their contents. So thank you so much again for joining me for today's lecture, and I wish you all a very good day. Thank you so much.